So as you know, this is a virtual conference, the hours 2021 due to current situation in the country. So pre-recorded presentations will be played. And please rename the presenters, please rename your Zoom window with your session ID so that it's easy for us to identify, right? So the presentation will take about 10 minutes. And altogether, we have 15 minutes. The second 5 minutes will be for questions and answers, right? So please keep all your microphones muted and video switch off, so that it's easy for the rest of the participants. But during Q&A sessions, so you can open your unmute and then you can answer the questions, right? So anybody who wants to answer any question, again, the same procedure. So again, there are 5 presentations lined up for the second session too. So let me begin with the first presentation, that is ID 122, a preliminary survey on species diversity and habitat distribution of amphibians in Gannoruwa Forest Reserve, Kandy, Sri Lanka. Authors are B. Pathirana, K.M.S. Ruvinda, T. Kotigala, K.M. Gunatunga, and S.B. Hera. The presenter will be T. Kotigala. Over to you.
This research study presents a preliminary survey on species diversity and habitat distribution of amphibians in Gannoruwa Forest Reserve, Kandy, Sri Lanka, authored by Buwanaka Pathirana, Sudesh Ruvinda, Tevuni Kotigala, Kanishka Gunatunga, and Sanath Bandara Herath. In the recent decades, tropical amphibians have undergone drastic population declines where nearly 41% of the amphibian species are threatened with extinction, making them one of the most threatened groups of animals on Earth. However, according to the data produced by the ScienceNet journal, a data deficiency is indicated for the status and threats for amphibians in Sri Lanka as depicted in the map. Sri Lanka is known as a biodiversity hotspot along with the Western Ghats of India. The amphibian fauna of Sri Lanka comprises of 120 species where approximately 90% of it is endemic. This graph depicts the species composition in different geographic zones in Sri Lanka, in which Central Hills is the relevant part for this particular study. Threats to amphibians, which are increasing at an alarming rate, remains a conservation concern. However, amphibian diversity, distribution, population viability, breeding biology, and behavior in isolated and anthropogenically impacted landscapes still remain a poorly addressed research focus in Sri Lanka. Gannoruwa Forest Reserve is a significant site where holotypes, paratypes, and neotypes of several amphibians and reptiles have been recorded. This study aims to deeply understand the amphibian diversity and distribution of this important yet inadequately studied area. Thereby, there are 2 main objectives for this study. Firstly, to assess the amphibian diversity, habitat distribution, and anthropogenic impacts, and then to recognize the threats to amphibians and the immediate conservation interventions required to conserve them and their existing habitats. In order to understand the study site climate, Gannoruwa Forest Reserve receives a mean annual temperature of 24.1 Celsius degrees and a mean annual rainfall of 2,131 millimeters. Moving on to the survey method, this survey was conducted from December 2019 to October 2020 using the Visual Encounter Survey technique. The survey covered both wet and dry seasons, detecting diurnal and nocturnal species. As indicated in the map, the survey covered 18 pre-marked transects in 6 major habitat types, which included naturalized abundant plantations, grasslands, mahogany plantations, nutmeg plantations, the natural forest on the summit, and swamp areas. The transects were selected equally based on the habitat types, that is 3 transects per habitat with roughly 50-meter length, and were surveyed with a team of 3 members. Moving on to the data collection, potential habitats of amphibian fauna, that is all microhabitats including rocks and ground, dead and fallen logs, dense bushes and grass patches, rock and tree crevices, leaf litter, and water bodies were thoroughly searched. Every individual specimen was noted and identified to the most specific taxonomic level possible using standard field guidebooks and expert reconfirmation on identification through digital photographs. Anthropogenic impacts were recorded during each visit through observations and informal interviews with the neighboring villages. For data analysis, the Shannon-Wiener Diversity Index, evenness, and the dominance were calculated for each habitat type using PARS software. Moving on to the study results and discussions, a total of 254 individuals of 13 Anuran species belonging to 4 families were recorded. The encountered species were Dattaphrynus melanostictus from family Bufonidae, Hydrophylax gracilis, Indosilvirana temporalis, Lanchanectus corrugatus, and Minervaria agricola from family Dicroglossidae, Polypedatus cruciger, Pseudophylotus cavirostris, Pseudophylotus fergusonianus, Pseudophylotus popularis, Pseudophylotus rus, Pseudophylotus schneideri, and Pseudophylotus zoro from family Rhacophoridae, and lastly, Aperdan obscura from family Microhylidae were recorded. However, Sicilian species were not encountered during the research fieldwork. Excluding Dattaphrynus melanostictus and Minervaria agricola, 84.6% of the recorded species were endemic. As the figure indicates, out of all the species, Pseudophylotus zoro was the most abundant in the sample. Only one record of Pseudophylotus cavirostris was seen in the abundant plantation, while Pseudophylotus fergusonianus was recorded only in the forest on summit. Hydrophylax gracilis, Indosilvirana temporalis, and Lanchanectus corrugatus were recorded only in the swamp area. Pseudophylotus zoro recorded the highest relative abundance across all habitat types. As indicated in the table, the highest level of evenness was found in the grassland, indicating a near equal distribution of abundance across the species, while the lowest level of evenness was indicated in abundant plantations. Mahogany plantation indicated the existence of a dominant species. Overall, swamp area indicated the highest richness, highest number of species occurrence, and highest diversity among all habitat types. The Shannon-Wiener Diversity Index, arranged in the decreasing order, indicates the lowest diversity in the mahogany plantation. Apart from this, previous studies and personal communication revealed that major threats caused by anthropogenic impacts result in habitat degradation for amphibians in Gunn-Oro Forest Reserve. Furthermore, alien invasive plant species can enforce a possible threat by absorbing water resources, leading to drastic degradation of breeding habitats of amphibians. Except in the buffer zone, there is a lack of a stable water source at the Gunn-Oro Forest Reserve, which may also seem to create an unavoidable problem for amphibians. Moving on to the conclusion and recommendations, this study provides foundational data that could be used to recognize local amphibian species distribution patterns in different habitat types in Gunn-Oro Forest Reserve. 2 species, that is Pseudophylotus zoro and Pseudophylotus rus, were found occupying all the 6 habitat types, and relatively high species diversity was observed in the swamp area. A higher percentage abundance of amphibian distribution was observed from 500 meters to 550 meter altitude, which may indicate a correlation with the existence of swamp area. However, further scientific research must be carried out to explore the existence of a correlation. Anthropogenic impacts, invasive alien plants, and climatic impacts causes habitat degradation of amphibian fauna. Therefore, this study recommends conservation decisions to incorporate amphibians and emphasize the importance of habitat heterogeneity in any future conservation initiatives for their long-term survival in Gunn-Oro Forest Reserve. These are the main study references among the resources referred.
Thank you
Thank
very
you.
much. All right. Thank you very much, Ms. Kottigale and the team for the presentation. It's very informative and very good initiative to study a new place to Gandarva Forest Reserve. Now the forum is open for relevant questions.
Yes, madam, can I ask?
Yeah, sure.
Yes, Termini is a good study and good presentation. My concern is that you have said that your amphibian diversity and evenness is most of the grasslands. But in your anthropologic activities, in your presentation, you are not indicating any thing about that grassland. If there is any the traits in the grassland in the Gannoru forest, or it's intact. Yeah. Hello.
Can I let Sudesh or Bhuvanika take that question if they don't mind?
Yeah, it's
Not
yeah,
yet.
it's okay.
Okay.
Yeah.
Sorry, sir, can you repeat the question?
Yeah, basically you said that the most of the amphibian diversity and the evenness is in the grasslands. But in your anthropologic activities, are there any anthropologic activities in the grasslands in
Yes.
that particular area?
Like setting fires and everything that destroys their habitats. I think that was mentioned in the study that they set fires to the grasslands for their cultivation work. So that in turn has an impact on the habitats destruction.
Yeah, those habitats mostly used for their breeding or the staying or something, the food sources or
both, I guess.
Yeah,
Yes.
both. They use both.
Okay, thank you.
Any more questions? I have a simple question from the team. Actually, you said the under mahogany plantation. I don't know how, because you once you said they are more concentrated to swamp areas, and then you are saying the dominance is more under the mahogany plantation. So it's sort of like any special the prominent species or something like that.
Yeah, the Pseudopilotus sauro is the prominent species, and among the mahogany plantations, I think 95% of Pseudopilotus sauro, and I think that the reason is the leaf litter under the trees are that leaf litter layer is it is a very suitable habitat for the Pseudopilotus. So I think that's the reason.
So is it a kind of small and creature, kind of size-wise smaller than the rest?
Yeah. All the Pseudopilotus species are smaller ones. That one also same size, but I think that habitat is very suitable for their behaviors. I think that's the reason.
Yeah, maybe because otherwise, as far as I know, usually the mahogany plantations are really dry soil. Yeah. So then, okay. Thank you very much for the very informative presentation, though it is very beginning like a fundamental study, but it's very useful for further studies. Hope you all will continue with this type of studies in the future. Thank you very much. Right. So let's move to the second presentation for today, for the second session. Bioactivity studies on crude extracts of fungi isolated from Cereopstagal and surrounding soil from Sri Lankan mangrove environment by WCCN Fernando, C. Ranasinghe, and MM Kader. So the presenter will be WCCN Fernando. Over to you, Mr. Fernando.
Good morning all, I am Chintaka Fernando. The topic of my presentation is bioactivity studies on crude extracts of fungi isolated from Cereopstagal and surrounding soil from Sri Lankan mangrove environment. This is the outline of my presentation. introduction to microbial natural products, isolation and morphological identification of fungi, fermentation of pure fungal cultures, extraction of fungal cultures, preliminary screening and TLC analysis of fungal extracts, bioactivity studies on fungal extracts, spectroscopic analysis of fungal extracts, and finally, the summary. First of all, I will explain what natural products are. Natural products are naturally derived compounds produced by living organisms such as plants, animals, and microbes. Among natural products, secondary metabolites are important for survival from others. As all we know, the story of the accidental discovery of penicillin. It was isolated from the fungus Penicillium notatum. This antibiotic saved lots of humans in the world and will save more. This discovery attracted the science community to work on microbes like fungi. For my research, I have chosen some endophytic fungi because they are reported to contain various types of active compounds such as anti-cancer, antibacterial, antioxidant, insecticide agents, anti-diabetic, phytotoxic, antifungal, etc. And also, endophytic fungi show a great potential for genetic modification and recombination with plant genes so that they can synthesize compounds for the host plant. For sample collection, I have chosen coastal area, the mangrove environment, the plant which adapt to high salinity, temperature variations. This is the plant Cereops tagal, the mangrove from which I isolated endophytic fungi. Previous studies have shown this plant to contain important bioactive compounds. Then, the isolation of endophytic fungi was carried out. First of all, the leaves were washed thoroughly with running tap water. Then they were triply sterilized. After the sterilization, the plant leaves were cut into small pieces and inoculated into freshly prepared PDA media and incubated at room temperature. After the 5 days, some of the growing colonies could be seen. Then they were serial transferred into PDA media and got 3 pure cultures. This is the procedure I have followed to isolate fungi from soil. 1 gram of soil suspension was mixed with 9 milliliter of distilled water. Then a dilution series was prepared until 10 to the power minus 6. They were serially transferred to PDA media, and after the 6 days incubation period, some of the growing colonies showed up and obtained 1 pure culture. Then the morphological identification of fungi using colony characters was carried out under light microscope. The 2 fungi, MPG1 and MPG2, were identified as penicillin species, another one as Silaria, and the last one is unidentified. Next, the large-scale fermentation of few fungal cultures were carried out. Few fungal cultures were grown in marine rice media in 12 flat bottles and incubated at room temperature for 30 days until fermentation. After the incubation period, the extraction of few fungal cultures were carried out using ethyl acetate. After the ethyl acetate extract was partitioned with hexane and methanol. These are the yields of the fractions obtained. Among them, G1 and G2, the 2 penicillium species, gave comparatively high yield. Then, analysis of fractions was carried out under 2 categories: chemical analysis and bioactivity studies. Under chemical analysis, preliminary screening for major classes of compounds, TLC analysis and spectroscopic analysis was carried out. Under bioactivity studies, DPPH antioxidant activity and Brainstorm assay was carried out. With the preliminary screening, all the fungal extracts showed that they contained phenolic compounds and steroids. For the phenolic compounds, ferric chloride test was carried out, and for the steroids, Lieberman-Bouchard reaction was carried out. TLC analysis was carried out using two visualization methods, one with UV light and other one was with anisaldehyde spray reagent followed by heating. TLC analysis of hexane fraction with eluent chloroform indicate it contained various types of compounds like this, and TLC analysis of methanol fraction also indicate various types of compounds. Under bioactivity studies, DPAPH antioxidant activity was carried out. The harmful high energy nitrogen and oxygen radicals can form during the metabolic reactions inside the body. Most of the natural products have the ability to reduce these harmful radical to less harmful compound. Antioxidant activity is reported here in terms of IC50 values. A lower IC50 value reflects a greater antioxidant activity of the sample. According to the IC50 values obtained, unidentified fungi indicated the maximum ability to reduce radicals compared to the well-known control ascorbic acid. Brainstorm lethality assay against Artemia salina was carried out to check the cytotoxic activity of fractions. MPG1 and MPG2 extract kill all the nuclei at the lowest concentration, it indicating it to contain high active cytotoxic compounds. After that, isolation of secondary metabolites from bioactive fractions of MPG2 was carried out. The three major secondary metabolites, pterine, imodine, and ergostrol, were isolated using HPLC. In this structure of pterine and corresponding HNMR and HSQC spectra, this is the structure and HNMR and HSQC for the compound imodine. This is the spectrum and structure of ergostrol. All the fungi contain ergostrol because it's one of the major compounds found in the cell walls of fungi. In summary, 3 endophytic fungi and a soil-associated fungus from mangrove plant and its surrounding soil were isolated. Crude extracts of all 4 fungi showed the presence of phenolic compounds and steroids. The fungus MPB1 showed a significant antioxidant activity. Extracts of all 4 fungi showed cytotoxic towards brainstorms. MPG1 and MPG2 contains potent cytotoxic activity compounds. Secondary metabolites of fungi isolated from Sri Lankan mangrove habitat contains compound with potent biological activities. This will give great opportunity to isolate compounds in larger amount in less time without disrupting the bioactivity. These are the references and acknowledgement, and thank you all.
Thank you very much, Mr. Fernando and the team. It's a wonderful, very good research, very good piece of work, and you presented very well as well. Now with the open for discussion, questions.
Yes, madam. Shall I ask one question?
Sure.
Yes, Mr. Fernando. Now you used 2 types of medium for culturing your fungus. Now for the inoculation, you use protect PDA medium, and then for your large scale extraction, you use, I think, rice medium. Now, have you find any difference in growth of fungus, or why especially you select rice medium for large scale fungal growth? Is there any special reason?
Yes, thank you very much for the question, Mr. Priyanta. Yes, actually, a very interesting question. The thing is, when we change the media, some of the researchers with the literature survey, we analyze some fungus, some of the fungi, they can produce more advanced compounds with the rice media rather than PDA media. And also we got some the plat bottles also available with our university lab. So we decided to go with the large scale for with the rice media, marine rice media. That's why, that's the reason.
Right. Okay, thank you, Chinta.
Any more questions from the participants? I have a small question from you that now you isolated, I think out of the 4 fungi you tested, I think one is from soil. So you did everything together, or you have any idea about the soil fungi contains more sort of like in your summary, the last sentence is the some sort of, can I go back to the last slide, please? The summary slide. Yeah. This will, the secondary metabolites of fungi isolated from Sri Lankan mangrove habitats contain compound. Is this that fungi? The soil one, or you are referring to all 4?
Yes, madam, mainly all 4.
All 4. You have no specific idea like how they differ from the other surface leaf, surface endophytes, and this the soil one. You know, any kind of like compound wise or anything like that.
Yes, madam, it indicate majorly the plant fungi contain high active rather than soil.
All the 3 compounds you isolated.
Yes, madam.
All right.
But soil one also, it contain active, but when we comparatively
They are more.
Yes, madam.
Yeah, and also sort of like can you relate this kind of substances you isolate from the mangrove environment like with the literature to the other similar organisms what you find on the other plants, like any difference due to the environmental conditions, or while you are reading, not during your research?
Yes, madam. You're asking for a specific mangrove environment.
Yeah. Why is it more certain compounds or like that under harsh environment where we are experiencing with mangrove?
Yes, madam. That's why, because of the fungi which contain with the mangrove plant, they should have the potent to avoid these harsh conditions. That's why all the compounds should
All these
consist.
metabolites are more with the harsh environment.
Yes, the adaptations. That's why, madam.
Right. Thank you so much for the very important presentation. Hope
Thank you, madam.
continue with this, and the team. Thank you so much. Right. So we'll move on to the next presentation. Right. Next presentation for today is awareness common zoonotic diseases among the new BSc registrants of the Open University Sri Lanka in 2020. The authors are V.S. Kulatunga, L.P. Fernando, U.A. Jayawardena, and C.D. Jayasinghe. Presenter will be V.S. Kulatunga. Over to you, Ms. Vidyana.
Greetings to everyone. I'm Vidyana Kulatunga from the Open University of Sri Lanka. Today I'll be presenting our study, Awareness of Common Zoonotic Diseases among the new BSc degree registrants of the Open University of Sri Lanka in 2020. What are zoonoses? Zoonotic diseases or zoonoses are a group of infectious diseases which transmit among vertebrate animals and people. They represent 60% of emerging infectious diseases all around the world. Though a part of scientific community is already aware of these zoonoses, the global attention was oriented towards this topic with the emergence of COVID-19, the global pandemic, which we are now experiencing devastating effects on public health and the economy in the world. Considering the zoonotic pathogen Zoonosis can be grouped such as bacterial zoonosis, viral zoonosis, parasitic zoonosis, etc. In 2017, Indian subcontinent was identified as a hotspot for emergence of new infectious diseases. Our country, Sri Lanka, was also at an elevated risk of emergence and re-emergence of viral zoonosis such as bird flu, rabies, and leptospirosis. So the significance of the study is related with the awareness of zoonosis in Sri Lanka. Major drawback towards the prevention and control of zoonosis is the lack of awareness. Therefore, raising awareness on zoonosis at all levels of society is important to reduce this risk. To initiate this, selecting a group of individuals is very important. University students are considered as most of the young population of a country with ample health literacy. Their level of awareness on zoonosis may imply the need for health education at the university level. When considering the student community of OUSL, it is large and spread throughout the country, and it is also supported with a number of regional centers and study centers of the university. This population also has a variety of demographic aspects such as occupation, age range, and their hometown. So, this cross-sectional study was conducted to evaluate the awareness, knowledge, and attitudes to common zoonotic diseases among the new registrants of the BSc degree program at OUSL in 2020. Moving to the methodology, we applied a cross-sectional study design to a cohort of new BSc registrants participated in the Empowerment for Independent Learning program held in December 2020 at OUSL. The study was conducted using a validated, structured, and self-administered online questionnaire that was emailed to students as a Google form. The questionnaire was consisted with 4 parts to collect information about demographic characteristics of the respondents, awareness on zoonosis and transmission, knowledge on types of zoonosis, and attitudes towards the prevention and control of zoonotic diseases. Then the collected data was analyzed using SPSS version 20 Pearson chi-square test. Coming to the results and discussion, the total number of respondents were 83. First, the demographic characteristics data are summarized here. Most of the respondents represented the age range of less than 24 years. 81.9% of respondents were female students. Marital status was single of the majority. 79.5% of the students were unemployed, and others represented occupations such as teaching, medical laboratory, IT sector, and agriculture sector. 95.2% students were enrolled to biological science stream, and the rest of the students were enrolled to computer science among the respondents. These data of demographic characteristics were later used to cross-relate with other aspects such as their knowledge on zoonosis, practices, and attitudes. When it comes to the awareness on zoonosis and transmission among the respondents, 83.2% of the respondents knew that zoonotic diseases are transmitted to humans by animals. 79.2% of them were aware that zoonotic diseases can be transmitted to humans by direct contact with animals. 85.6% of them knew that zoonotic diseases can be prevented. Only 67.5% of the respondents knew that zoonotic diseases can be controlled, while 86.8% of respondents was aware that zoonotic diseases can be treated. These results indicate that most of the respondents had a satisfactory level of awareness on zoonotic diseases. The proportion of the respondents who recognized the diseases included in the questionnaire zoonosis are variable. The questionnaire consisted with 28 viral, bacterial, and parasitic zoonoses. Highest recognized zoonotic disease was COVID-19, and it was followed by swine flu, brucellosis, SARS, and rabies. The least identified zoonotic disease was hepatitis E. When relating demographic characteristics with awareness of zoonotic disease types, a higher proportion of female respondents than male respondents identified bovine tuberculosis as a zoonotic disease, which is a bacterial zoonotic disease transmitted to humans by cattle and other wildlife. The same disease was also recognized by students enrolled to biological science stream than computer science students among the respondents. This could be due to the knowledge they have obtained during their secondary education. Q fever is another zoonotic disease caused by a pathogenic bacteria, and it was recognized by a majority of female students than male students among the respondents. Swine flu and SARS was identified as zoonotic diseases by a higher proportion of students who had age range less than 24 years. The knowledge on different types of zoonotic diseases were related with the pet owners among the respondents. Interestingly, brucellosis, rabies, HIV, cryptosporidiosis, toxocariasis, giardiasis, and cysticercosis was identified by more pet owners. Pet owners appear to be educated by veterinary practitioners or from other sources. When considering the awareness among respondents on attitudes and practices to prevent zoonotic disease transmission, the highest recognized practice was avoiding direct contact with animals, with a percentage of 91.1. The other practices such as avoid eating raw meat, avoid eating unwashed fresh fruits and vegetables, avoid rearing pets in indoors, ensuring the regular vaccination of pets, and washing hands thoroughly after petting animals were also at an satisfactory level. A significant higher percentage of female respondents agreed that zoonosis can be prevented by avoiding eating raw meat and avoiding eating unwashed fresh fruits and vegetables. However, it is inferred that students can be more educated about preventive and control measures of zoonotic diseases. Concluding the study, it was revealed for the first time that the BSc registrants of OUSL were satisfactorily aware of zoonosis. The outcome of the study may have implications to initiating awareness programs on preventive and control measures on zoonosis among the BSc students at OUSL. It is warranted to extend this study to a larger population of students to obtain a broad understanding of their awareness. These are our references. We would like to give our humble acknowledgement for the support given by Mrs. Nalini Edrisinghe and Mr. D.L.N. Jayatilaka from the Open University of Sri Lanka. And thank you, everyone.
Thank you very much, Ms. Kulatunga and the team. Right, it's very nice to listen to your presentation after going through several drug discoveries and very high experimental research. And it's very happy that it's a totally the survey type questionnaire used, and then you have commenced this kind of activity, I think, for the first time in the OUSL. Right, right. It's open for discussion now. Any questions from the participants?
Yeah, I'm Chandani here. This is a very interesting topic, actually, as you said. And I'm just wondering, was it accidental that you had missed out physical science students, or why was it? Because you are referring to the awareness among BSc degree registrants, and I would imagine perhaps physical science students, their awareness could be perhaps different to bioscience students.
Madam, thank you very much for the question.
Well, we applied this questionnaire for all the BSc degree registrants, but voluntarily they responded only 83. Among them, part of them responded as their stream as computer sciences. I think they comes under physical science.
Yeah, in such a situation, I would not perhaps refer to this as among the BSc degree registrants, because we have to be more careful when other students are not represented. Whether you can say something about them is questionable.
I think, Dr. Navaratna, they are totally 692, I remember. And then they have got the replies only from 89. So then that could be the main reason, because they have sent it to 692. So because of that, most of they are, in the sense like the secondary education, all those have come into effect because of the having more students, more unemployed people, and less than 25. All those are factors, I think, that towards the biasness of the whole results. I think they have said in the summary that they will be going for a large number study. So
I agree. That's really good. But I'm suggesting it's safer to write the conclusions accordingly. You know?
Okay.
Professor Tamara, I'm Dr. Chanika. May I add something to the Professor Navaratna's question? Actually, madam, we employed it so without any biasness to all the students, but only the biological sciences, the one who responded to the questionnaire. So it was difficult to get the response from the physical sciences because maybe they were not interested by looking at the questionnaire. But then again, we employed the questionnaire again through the emails by like encouraging them to reply, and then after that we got several other responses also, and I think we are hoping to write a full paper based on this one. So later on we got several responses from the physical science students as well.
Yeah, thank you.
Okay, thank you. Just like, were they different to the other students? Oh, you have perhaps not yet looked at them, no?
I think we have looked at, but it was not different from what we got from this 83 the participant. They also had a significant knowledge on the zoonosis, but not about the specific diseases, but the general stuff they were aware, madam.
Okay, thank you.
Or else, Dr. Chanika, you can call this as because since the number happened to be large with bioscience students, or later you can change if you are going for a paper, because as President Dr. Navaratna said that there's an implication due to when you say BSc registrants, of course you can either add other participants in the future, or you can just rename, because most of the information I think
exactly, madam. Based on the population.
Yeah.
I think Tamar, one correction. I'm not professor, but anyhow, now you put it in the right way. I was trying to suggest that you change the title, and perhaps you can strengthen the discussion to say that you attempted, but they had not responded. But anyway, I was just saying I'm safer to change the title, or perhaps the conclusions. Even at the end, you had said no BSc registrants. Fine, thank you.
Exactly, madam. Thank you very much for your valuable comment, madam. Thank you very much.
It's very interesting research. Please proceed with that. Okay, let's move to the presentation, right? Growth and yield performance of rice or Rhiza sativa varieties under nano micronutrients fertilizer, nano copper oxide, and nano zinc oxide. Authors are CLT Sandanayaka, SR Veerakone, N. Karthikeyan, S. Somaratna. Present will be CLT Sandanayaka. Over to you, Ms. Sandanayaka.
A good day to you all. I'm Chamodi Sandanayakar, and this presentation is about growth and yield performances of rice varieties under nano micronutrient fertilizers, nano copper oxide and nano zinc oxide. Rice is a semi-aquatic plant of family Poaceae which comprise 22 species. Rice cultivation began about 8,000 to 15,000 years ago. In the present, the ecological varieties Indica, Japonica and Javonica are cultivated in different regions of the world. Today, rice is the second largest commodity produced worldwide and it is the staple food of 3.5 billion people around the world. During the last decade, the top rice producers were China, India, Indonesia, Bangladesh, Vietnam, Thailand, Myanmar, Philippines, Brazil and Japan. Rice has a rich content of carbohydrates, lipids, proteins, fibers, water, vitamins and minerals, and therefore rice is considered as a potential carrier of nutrients for micronutrient fortification in humans. Micronutrients are essential in normal plant growth and development. These micronutrients can enter the plant either through roots or through the aerial parts. Zinc and copper are two such important micronutrients for rice growth. Immobilization of these micronutrients as well as zinc and copper deficiencies in paddy soil are well reported. Copper is important for photosynthesis, mitochondrial respiration, carbon and nitrogen metabolism, oxidative stress protection, cell wall synthesis, redox agent, and a catalytic metal. Zinc is important for chlorophyll production, maintain membrane activities, cytochrome and nucleotide synthesis, and for development of seeds and stalk maturation. Depending on the rainfall, Sri Lanka has three agroecological regions as wet zone, intermediate zone, and dry zone. Soils in dry zone and intermediate zone, which are major rice growing areas, have reported to be deficient in micronutrients, which reduce the crop yield. To compensate the nutrient loss in soil, farmers are advised to add chemical fertilizers in recommended amounts. This include the main three types of fertilizers for rice: urea, triple superphosphate, and muriate of potash, and also the micronutrients such as copper and zinc. Although there are fertilizer subsidies, the global price of fertilizers increases, and in many instances it has been reported that many farmers apply fertilizers less than the recommended amounts, which eventually decrease the rice production. Nanotechnology is a novel and innovative tool that has been very useful in the field of agriculture. The US National Science and Technology Council states, the essence of nanotechnology is the ability to work at the molecular level, atom by atom, to create large structures with fundamentally new molecular organization. Nanoparticles show different behaviors than their bulk particle form. Nanofertilizers have large surface area and particle size less than the pore size of leaves of the plant, which can increase the penetration into the plant tissues from applied surface, and this improves the uptake and nutrient use efficacy. Nanofertilizers, when applied as foliar sprays, can enter the plants through stomata and travel through the plant in apoplast, symplast pathways and through the plasmodesmata. Therefore, this research study was carried out to study the nanofertilizer effect on rice and to study the feasibility of reducing the overusage of bulk fertilizers by introducing the nanofertilizers. This is how this study was done. 2 traditional and 2 inbred rice varieties of 3.5-month growth period was selected. 2 weeks old germinated seedlings were transplanted into pots containing paddy soil with recommended amounts of NPK fertilizers. Then the nanofertilizers were synthesized. First, thermogravimetric analysis was performed for the precursor chemicals, and then copper oxide and zinc oxide nanoparticles were synthesized. To confirm the particle size and shape, X-ray diffraction and scanning electron micrographs was done. Then the synthesized nanofertilizers were used to prepare suspensions at 4 different concentrations as 0, 30, 60 and 120 milligrams per liter in nano-copper oxide, nano-zinc oxide and the composite of nano-copper oxide and nano-zinc oxide. After that, the prepared nanofertilizers were applied during the bearing stage and during grain filling stage. The growth parameters were recorded at 30, 60, and 90 days after sowing. At the harvest, the yield parameters were recorded. Finally, the collected data was subjected to descriptive and inferential statistical analysis. Now let's move to the results of growth parameters. Here you can see the mean number of tillers with different treatments of nanofertilizers in 4 different rice varieties. But the mean number of tillers were inconsistent with the different treatments of nanofertilizers in all the 4 rice varieties that were tested. Here you can see the mean plant height with different treatments of nanofertilizer in 4 different rice varieties. For both traditional rice varieties, the highest mean plant height was reported in nano zinc oxide 60 mg per liter and nano copper oxide 120 mg per liter. For both inbred rice varieties, the highest mean plant height was reported in 120 mg per liter in both nano copper oxide and nano zinc oxide. Here this graph depicts the mean chlorophyll content index with different treatments of nanofertilizers in 4 different rice varieties. The mean chlorophyll index increased with the application of nanofertilizers. The mean number of leaves with different treatments of nanofertilizers in 4 different rice varieties are depicted here. As you can see here, the mean number of leaves increased with the application of nanomicronutrients, but the optimum concentration varies with the variety and the nanofertilizer type. This ANOVA table of plant height suggests that the plant height is significantly affected by the nanofertilizer, concentration, rice variety, days after sowing, and the interactions between those factors. This ANOVA table of chlorophyll content suggests that the concentration, nanofertilizer, variety, days after sowing, and some of the interactions between those factors have a significant effect on the chlorophyll content. Now let's move to the results of yield parameters. The mean number of panicles per plants, as you can see here, has increased with the application of nanofertilizers, and the optimum concentration was 60 mg per liter in all 3 nanomicronutrient fertilizer type in all 4 different rice varieties. As you can see here, the optimum concentration was 60 mg per liter in all 3 nanomicronutrient fertilizer types in all 4 different rice varieties for the mean panicle length with different nanofertilizer treatments. The mean number of grains per panicle has increased with the application of nanomicronutrient fertilizers, and the optimum concentration was 60 mg per liter in all 4 different rice varieties. The mean weight of 100 grains also increased with the application of nanomicronutrient fertilizers, showing optimum concentration at 60 mg per liter in all 3 different nanomicronutrient fertilizer types in all 4 different rice varieties. This ANOVA table of the number of panicles per plant suggests that the variety, nanofertilizer type, and the concentration of nanofertilizers had a significant effect on the number of panicles per plant. Here we can see that the variety, nanofertilizer type, concentration, and the interactions between those factors had a significant effect on the panicle length of each rice variety. Number of grains per panicle was affected by variety, element, concentration, and the interactions between these factors. Here we can see that the variety, nanomicronutrient fertilizer type, concentration, and the interactions between those factors had a significant effect on the weight of 100 grains. Finally, a principal component analysis was carried out, and the results suggest that the interactions of nanofertilizers are different from traditional rice varieties and inbred rice varieties. In conclusion, nano copper oxide and nano zinc oxide has a significant effect on plant height, number of tillers, chlorophyll content, and number of leaves. Nanofertilizer treatments can be used for efficient application of micronutrients into the plants, and inbred rice varieties and traditional rice varieties respond to the nanofertilizers in a different manner. These are the references that are used in this presentation.
Thank you very much.
Thank you very much for your kind attention and valuable time.
Thank you very much, Ms. Sandanaik and the team. Now the presentation is open for discussion. Any questions from the participants? Ms. Sandanaik, I noticed somewhere in your methodology, the write-up, you have said that you applied the nanofertilizer in 2 stages. One is at the bearing stage, one is at the milking stage, right?
Yes,
have you considered in your results? I didn't see any such stage difference, whether you considered both stages together or
yes, madam, we considered both of these stages together because at these stages, it is said that in bearing stage and milking stage, they take up more nutrients to produce the panicles and to produce the seeds. Therefore, we considered both of these stages together.
But the days are different. One is 48 to 58 days, other one is the more than 100 days. So
yes,
I don't know whether you can, it's sometimes if you have considered them separately, results would have been different with the applications, right? And that is a good presentation actually with good data and with good interactions, and then there is a way forward. And also just a small question that you have said the inbred and traditional varieties in your regression analysis that they are different. Any reason for that?
Madam, inbred rice varieties are hybrids, so they are more chemically intensive. They take up more chemicals, and they are like naturally they produce a lot of grains. So that may be the reason why they react a bit differently. I mean, even the tillering stage, we could see that inbred rice varieties, the tillering was a bit later. I mean, tillering happened later than the traditional rice varieties. So that could be a reason.
Yeah, that's good. That's the answer, but you should have incorporated that in your presentation, then it will be different. Okay, thank you very much, Ms. Sandanaik and the team. It's a very good research, right? Let's move to the last presentation for today's second session, right? The behavioral response of the red-wattled lapwing Vanellus indicus in different habitats. Authors are T. Galappathy, UKGK Padmalal, and TSP Fernando. The presenter will be T. Galappathy. Over to you, Galappathy.
The topic of my presentation is the behavioral response of red-wattled lapwing Vanellus indicus in different habitats. Urbanization is a trending topic on a global scale and the leading anthropogenic cause of habitat destruction. Sri Lanka is a part of the biodiversity hotspot of the Western Ghats and follows the global trend of urbanization, with many of its major cities becoming swiftly urbanized in the past decade. For instance, the following images show the rapid urbanization of the city of Colombo from 1995 to 2017. When a species faces urbanization, there are 3 possible outcomes: migration, local extinction, and adaptation. The novel urban ecosystems consist of species that can tolerate and adapt to the effects of urbanization. Bird species residing in a habitat are known to show adaptability towards it, as they are capable of migrating to a suitable habitat with ease if necessary. And presently, the study of urban species remains at rudimentary level in Sri Lanka. The red-wattled lapwing, a common vader, was chosen as a model urban species for this study, as it is widely distributed throughout the island in a variety of ecosystems. The understanding and conservation of waders aids in the understanding of wetlands and the residing bird species, which will in turn pave the way for future urban conservation efforts of Sri Lanka. The aim of this study is to aid future urban conservation efforts in Sri Lanka through the investigation of a species, the red-wattled lapwing, that is well established in an urban environment, and to investigate to what extent is urbanization detrimental to a species. The objective of this study is to evaluate the behavioral response of the red-wattled lapwing in different habitats: urban, semi-urban, and natural. Moving on to the methodology, 3 study sites were chosen for the behavioral study of the red-wattled lapwing: the Open University Naval premises as the urban study site, Polga Sovita as a semi-urban study site, and Anavilundava Sanctuary as a natural study site. 3 microhabitats were selected for each study site, and a microhabitat was defined as a subset of each habitat, which are displayed as follows. The following methods were used for the behavioral study. Ad libitum sampling was conducted in each study site, and the behavioral states and events of the red bottle lapwing were identified and defined. A pair of adult red bottle lapwings were ringed at each microhabitat using the misneck technique with the aid of a color-accurate decoy and a playback call. The behavior was recorded at each microhabitat at 10-minute intervals from 6 a.m. to 7 a.m., 12 p.m. to 1 p.m., 5 p.m. to 6 p.m. The frequency budgets and time budgets were created for each microhabitat. The study was conducted for a total of 42 hours at the Open University Naval premises, 36 hours at Polga Sovita premises, and 27 hours at the Anavilundava Sanctuary. The data obtained was statistically analyzed using IBM SPSS version 20. A non-parametric test of Kruskal-Wallis was performed on the frequency data and time budget of the behavioral states of 3 study sites. And finally, the mean standard deviation and significance of the behavioral states were obtained. Moving on to the results. When we consider diurnal variation versus the mean frequency of behavioral states, the mean frequency of behavioral states of resting, foraging, and locomotion showed a significant difference among the 3 study sites and are defined as follows. This illustration depicts the events of the behavioral states of resting, foraging, and locomotion, respectively. When we focus our attention to the foraging behavioral state, significant differences were observed in the feeding behavior between the diurnal variation across the 3 study sites, primarily in the morning and afternoon hours, as seen in the table. Birds at the natural study site showed higher feeding behavior compared to the birds at the urban study site. Birds in the semi-urban study site showed the highest foraging behavior during the morning and evening hours. When we consider the locomotion behavioral state, significant differences were observed in the morning and afternoon hours of the natural and semi-urban study sites as seen in the table. When comparing the evening and afternoon hours of the urban study site and the semi-urban study site, the afternoon hours showed a reduced locomotion behavior, with the semi-urban study site depicting the least locomotory behavior. Considering the resting behavioral state, significant differences were observed in the mean frequency of resting behavior between the diurnal variation across the 3 study sites, primarily between the data obtained from the Anavilundava and Open University study sites, as seen in the table. An ascending trend of resting behavior was observed between the study sites of Anavilundava, Polga Sovita, and Open University premises, respectively. A high resting behavioral state was observed in the birds dwelling in the urban habitat in comparison with the natural habitat. And birds residing in the semi-urban habitat showed similar resting behavior to the birds dwelling in the urban environment in the afternoon and evening hours. When we proceed to the diurnal variation versus mean time of behavioral state, among the mean time spent on various behavioral state, only time spent on foraging showed a significant difference among the 3 study sites. It was also observed that the majority of the time was devoted to resting behavior in all 3 study sites as shown in the graphs below. Birds in the natural and semi-urban study sites devoted more time for foraging and locomotion activities when compared to the urban study site. When considering the time budget for foraging behavioral state, high foraging behavior was observed throughout the day in birds residing in the natural study site as shown in the graph. A descending trend of time spent on foraging was observed in birds residing in the natural, semi-urban, and urban study sites. Birds in the urban study site were observed to have minimum foraging activity. Nests are usually constructed on the ground with dry leaves and twigs, and at times with small stone pebbles as substrate depending on the surrounding surface. A special observation of a nest in the urban study site was observed to be constructed on a concrete surface with small stone pebbles as a substrate in very close proximity to a construction site. The red bottle lapwing was also seen to occupy rooftops of buildings at the urban study site periodically. Moving on to the discussion, several studies have shown that the sedentary nature of species have provided the opportunity for the establishment in urban environments, which can be attributed to the red bottle lapwing. When we focus on the foraging behavior, the results from the behavioral study of mean foraging frequency depict a high level of feeding activity in the morning, a decrease during the midday, and a lesser increase during the evening hours within all 3 study sites. This diurnal variation in a single study site could be attributed to the temporal changes in prey abundance and the inability to dissipate sufficient heat via evaporation and normal breathing during high temperatures of the midday, which leads to shade-seeking behavior during midday. Among the 3 study sites, birds residing in the urban environment have shown to spend less time during the day foraging and display reduced frequency of mean foraging behavior. Some studies have shown that arthropods are found more abundantly in urban gardens more than forest areas. The availability of food resources in urban habitats have played a key role in the reduction of mean frequency and mean time spent on foraging activity in the urban habitat. Some studies have shown that the artificial illumination in urban areas has made the nocturnal preference index higher for betta species, which may also account for the reduction of the mean foraging time and mean frequency of the red wattle lapwing in urban study sites. In contrast to the urban habitat, the mean foraging behavior of birds in other study sites, semi-urban and natural, was high. The reduced time spent on foraging by red wattle lapwing in semi-urban environment compared to the natural environment corresponds to studies that have shown a higher abundance of insects in small patches of vegetation in comparison to larger ones. Proceeding to locomotion behavior, interestingly, the mean time spent on locomotion saw an ascending trend of birds in the urban, semi-urban and natural study sites in their respective order. The red wattle lapwing in urban areas showed low mean flying frequency when compared to the semi-urban and natural study sites, and they showed tolerance to the presence of humans and human-related activities. This corresponds to studies that depict that urban birds are found to be more tolerant of humans than their rural conspecifics. The highest mean flying frequency in semi-urban habitats during morning and evening could be explained using the above due to low tolerance of red wattle lapwings to humans engaged in agricultural activities. High mean locomotion frequency was observed during the morning, which could complement the high foraging activity during the same time period by means of food searching. Looking into the resting behavior, an ascending trend of mean resting behavior was observed in the natural, semi-urban and urban study sites in their respective order. Elevated levels of mean resting behavioral events such as preening and tail jerk was observed in birds residing in the urban and semi-urban habitats, which shows that there is a possibility of the birds dwelling in these habitats to have a higher number of ectoparasitic infestation. Among the three study sites, a large share of time was devoted to resting behavior in the semi-urban and urban study sites in the afternoon and evening hours. This corresponds to the findings that exhibit behavioral changes in endothermic animals in the presence of high temperatures, such as reduced activity. The construction of a nest on concrete surface has been found to reduce incubation time. The presence of red wattle lapwings on rooftops of buildings has been observed in some studies, and it can be viewed as an adaptation to urbanization. The management of urban environments with green spaces and green roofs may aid in the future conservation efforts of species residing in urban areas. The study concludes with an indication of a significant difference in the behavioral patterns of foraging, locomotion, and resting between the study sites of Open University, Polka Soviet, and Anandundao, Sri Lanka, thereby concluding that the model species of red wattle lapwing has shown certain adaptations to a continuum of different habitats, urban, semi-urban, and natural. Further studies on the comparison of the physiology, pollution parameters, and parasitic load of species will give a comprehensive picture of a model species dwelling in the urban habitats of Sri Lanka.
Thank you. Thank you very much, the team, for the interesting presentation that is all about this, how these natural habitats are being changed by the anthropogenic activities and how the birds are really reacting to those things. And they have taken very interesting 3 ideas. Right, it's open for discussion now. Any questions from the participants?
I'm Naina here.
Yeah.
Mr. Shan, you have observed breeding only in the OUSL study site. So it means there is a difference in the time period you have observed, no? And maybe that have affected your results as well. What do you think?
Madam, we did not include the breeding data in this study, in this presentation, because we had several gaps in data collection due to COVID, because we couldn't go visit due to the restrictions of interdistrict travel and certain other things, like a certain region of the Anandundao forest caught in fire. We had certain political problems relating to the clearance of land. So the breeding data was not included in the study. We only measured non-breeding data such as foraging, locomotion, and resting behavior.
Okay, thank you.
Yeah.
Thank you, madam.
Can I add something on that, madam? Yeah. Basically, madam, in the urban setup, the gestation period is lesser compared to the other sites. Therefore, normally in the urban setup, we have seen a lot of pups in compared to the forest areas. I think the Prashant find those kind of results, no? But
yes, sir.
Yeah.
Okay, right.
Thank you, the team and the team, and it's an interesting presentation. Just for my curiosity, just Anna Vilundava, I was expecting a kind of like totally natural habitats. As you said, now there are anthropogenic activities, clearing and all that like. So I expected it to be kind of control and to see the rest how they are changing. But from your results, I see like even their behavior even under natural conditions is also changing. Is that so? Like, is it correct what I got?
Madam, our study sites were inside the sanctuary, but the clearing and the forest fire was actually, the forest fire was within the sanctuary, but the clearing was within the borders, like near the border, like buffer zone of the sanctuary. But when those problems occur, it is not safe for us to actually go and collect data. That is the reason we couldn't collect the data as mentioned for the breeding season of the lapwings. But the non-breeding season, we managed to collect within the sanctuary, which has minimum disturbances.
Because things like now the foraging, like they don't have to do any changes, no, like they are really in their own habitats, like they are adapted to their things like that is what I thought. This is just for this thing. Okay, thank you very much for the nice presentation. It's very informative. Hope you all will continue with the same thing. So that is the end of the session 2, parallel session 2, the biological sciences. So let me thank all the presenters and the participants and the authors for their very important, very good contribution towards ours 2021. I hope you all enjoy by exchanging ideas and meeting new people and things like those, reading new research findings and things like those. So thank you very much once again for all the participants and the technical team and the host and everyone. And it's a real success. Thank you very much.
Dear Professor Tamara, as the team convener, I would like to thank you for accepting our invitation to be the chairperson and conducting and managing the session very well with your vast knowledge in the field of biological science. And also I especially thank the technical team, Mr. Taranga and Mr. Salia, for your invaluable assistance.